several colleagues and collaborators, not only from India, but from China and also from many African countries. So I would like to welcome you all for this webinar. And before we start, I would like to congratulate Dr. Radhakrishnan and all the colleagues, staff, scientists, students of DGR for this 41st Foundation Day. So with these words, now I would like to share my screen and let me see how to open, because this is always some problem. Okay, I cannot do. So Radhakrishnan sir, you also need to do something for that sharing.
I'm trying for that. I'm on it. I'm on it. Kindly continue. I'm on it.
Okay, but I am trying to open my Nilesh. I think it's difficult to share that PowerPoint presentation.
You are allowed to share, all participants, all panelists.
How to do this thing? This share.
You see, sir. You see, it must be on.
Where is the presentation? There is no presentation. I already have opened this presentation. So this my presentation is open here. Sir, can you also allow me to share my presentation?
Sir, it is allowed, sir. Please see. It's allowed.
Now, this is not there. So when I go to the share.
Are you able to now?
No, this. Okay, now I can do it. Yeah.
Yes,
okay.
yes, sir. You can. Please go ahead, please.
Okay, thank you very much once again. So yeah, so I wanted to greet and congratulate all the colleagues and awardees today. And I was very excited to participate in morning session as well, where we had a presentation from Dr. T.R. Sharma, DDG Crop Science of Indian Council of Agriculture Research. So we were really heavily benefited. And I would like to thank once again Dr. Radhakrishnan for giving me this opportunity to share some of our work. Now, as I said, so once congratulations, and this is the 41st Foundation Day. And one thing that I know that all of you who are from India, they already know the role of DG. If not, especially for other colleagues, and I'm very happy to mention that DGR has been at the forefront of delivering outstanding research outputs, and they have developed and released several varieties and for different traits. So I think this is one of the major contribution of DGR. But in addition to that one, and I always feel very happy to, because I know Dr. Radhakrishnan for a long time. When I joined ICRISAT way back in 2005, and I mentioned in the morning, from that time point, I started to collaborate with Dr. Radhakrishnan. So Radhakrishnan believes very strongly in walking the talk, and he has been working not only in the research community, but the industry. And here you can see that how hard he is working in both directions, that so that we can have the better commercialization of the peanut varieties or groundnut varieties. He believes very strongly in that capacity building of the national stakeholders. Dr. Radhakrishnan and his team has been working very hard with these farmers, seed producers from the different parts of the country. So we feel very happy about you, Dr. Radhakrishnan, and I would like to once again appreciate all your efforts. The other thing which I want to mention and about the collaboration. Nowadays, collaboration is the key for any outstanding output or delivery. And ICRISAT and DGR has been collaborating for many, many years, from the time of our groundnut breeder, Dr. Nigam, then Dr. Janila, and also at the DGR, we had collaboration with several directors, including, I know 3-4 name only, but basically like when I joined at that time point, Dr. Muralidharan, Dr. J.B. Mishra, and now Dr. Radhakrishnan. And so, I mean, we are having really fantastic collaboration. Here you can see some picture of one of our former DG, David Worthington, with Dr. Radhakrishnan. And I have been visiting several times. And here Ratan Kumar and Radhakrishnan, and they are also coming to ICRISAT. My colleague Janila, groundnut breeder, my colleague Dr. Manish Pandey, genomics scientist. And these things have been really very, very successful. And we also have recognized the contribution of Dr. Radhakrishnan in early February or March this year as well for all his collaboration and contribution. And here I would like to show this picture. And I think you can see my cursor on the left hand side. This is Rajiv Varshney. On the right hand side, this is Radhakrishnan way back in 2008. And this was the
Rajiv, sorry for interrupting. The issue regarding the connectivity for the number of participants has been solved.
Okay, good. And you can see my screen, sir, right?
Yeah.
Okay, good. So here you can see that at that time point we were, well, you can see Dr. Radhakrishnan remained the same handsome and trim and slim person, but this guy Rajiv Varst on the left hand side kept on changing his phenotype. But well, that's the life. But nevertheless, what I want to tell that because of this collaboration which we started way back in 2005, we have wonderful collaboration output in different areas, including gene discovery and markers, molecular breeding products, and nowadays we are moving related to new area of the genomic prediction. And I will not go in the details, but I would like to mention that in addition to the varieties, which my colleague Janila is leading efforts on this direction and many other technology, pathology, etc. I'm just focusing here on genomics and molecular breeding aspect. And we feel very happy to have the collaboration with the DGR here in India. And while working with him, we also have very successful collaborations in China, in United States, and Africa, and I will share some of those results in this coming slides or so. So again, what I want to tell that because of all these collaborations, international collaborations, the Groundnut community has moved ahead. So now the question is that, well, what we need to do. But before we ask this question that for Groundnut, let's go back a little bit to one step back that why we need to be worried about all these things. Well, there are several international challenges at the global level which are related to the food and agriculture. And as we know that right now we are having around 7 billion or 7.23 billion population or something like that one. And this population is a result of the whole civilization by now. But interesting thing is that one third of this will related to about 3 billion population will be added in next just 40 years. Can you believe it? Anyway, and if you see that majority of the population addition is happening from Asia and from sub-Saharan Africa. And as a result, we need to provide the food for all of them. In India, so far, we are always saying 1.3 billion population, but recently I was seeing the data and now this is not 1.3, my friends, this is 1.4 billion. So this is an alarming point. And more than that, if we compare that right now, China is the most populous country, India is second, but in the 2050, India will take over and India will become number one, which is not good. And for doing this thing, we need to feed the poor people and forget the future even at present. We have these two issues are very, very important, hunger and malnutrition. Around 800 million people, they do not have the food to live a healthy life. And about 1.9 billion people, they are suffering from that malnutrition. This can be overnutrition or undernutrition. And on top of that one, and as I said earlier, if you see this world hunger map, 1 out of 9 people is going to bed hungry, which is not good. And this situation is much more serious in Africa and Asia continent. So we need to do something in this regard. Second is about the nutrition. So not only the energy or the food, but nutrition also play a very important role. And right now we are having these sustainable development goals. And for achieving majority of the sustainable development goals, nutrition is very, very important. And another thing is the climate change. So I was talking about the population, hunger, nutrition, and climate change. Unfortunate scene is that developing countries will be adversely affected from the climate change than the developed countries. And majority of time, the agriculture is the mainstay for the population, and we are dependent on that rainfall, etc. So I think that coming years are going to be much more challenging. So to address these issues, several international organizations like CGIAR and ICRISAT is part of the CGIAR. They work with large number of international organizations and national programs in Asia and Africa around the world, and they try to contribute in this particular direction. So many of you already are aware about the CGIAR, and here is the picture of the CGIAR that how the CGIAR institutes are spread over the world. And I'm coming from ICRISAT. So ICRISAT, our headquarter is here in India, but we are having our research operations in Ethiopia, Kenya, Malawi, Mozambique, Zimbabwe, and also Nigeria, Niger, and Mali. And I'm also very happy to see several colleagues participating from these countries in this seminar. So welcome once again. And of course, the other countries as well. So, and second thing is that, well, at ICRISAT, we work on the 5 important crops. And these are the basically 3, well, now we can say 6. So 3 cereals, 3 legumes, and I'm talking about this peanut. And so basically the groundnut or peanut is one of our important crop about which I'm going to talk in few minutes or so. Groundnut is very important crop from the protein perspective as well as from the oil crop perspective, and very nutrition rich food crops. And nowadays you have the different kind of cuisines and the different kind of diversity in terms of that getting those things. So I mean that ready are you to have products also. So I think peanut or groundnut is playing very important role. But now if we address the issue that, well, how we can enhance the crop productivity, how we can enhance the nutrition, so what we need to do in this context, all different disciplines of agriculture play very, very important role. However, in this presentation, I will be giving little bit or talking more about the plant breeding and genomic innovations, but nevertheless, we need the holistic view, and I will also discuss in few minutes about that. So let's talk about the plant breeding. Well, plant breeding is old science, and during the plant breeding, what generally we do that we try to address different issue. One is that we stabilize and increase the yield, and we try to address the issue of the pest and the disease resistance, and also maximize the resource use efficiency or water use efficiency. So this is one of the important thing. So this is the way that plant breeding has been very successful, doing great work. Now, what kind of milestones we had in the plant breeding? So we always talk about the innovations. In fact, there may be many, but in my opinion, or I would like to mention a few, one of them is the crop domestication, which happened about 10,000 years back, that across these different crops in the agriculture, we had the domestication. And after that, we started to have basically that at that time, this was very simple that if you need more crop, you just increase the area, you will get the more produce. But then in terms of the science-based plant breeding, I think this happened after the Mendel's law in 1865, when we had this, when Mendel's gave this law. And after that, I think in early 20th century, plant breeding started to happen based on the cross breeding. Then we started to have the hybrid breeding, and then we had this mutation breeding, etc. In 1953, we had the understanding of the structure of the DNA, and this changed the whole scenario of the modern plant breeding. And now when I say modern plant breeding, this may include molecular breeding, marker assisted selection, this may be the GM breeding, this can be the targeted breeding. So basically we started to have the breeding based on the genomic information. When we talk about the genome or genomics, so basically this in terms of that genomics, things started from 1990, and in the context of the human health, United States started a project called Human Genome Project. This took about 13 years and costed about $3 billion. And after 13 years, we had that first version of the human genome. And then human community or medical community start to think, well, we cannot afford $3 billion every time. Can we reach a stage that we can have one human genome in just $1,000 and just half an hour time? And because of this work, there were a lot of advances in the sequencing technology. Many centers around the world, they started to work in this direction. Even at ICRISAT, way back in 2007, we started the Center of Excellence in Genomics and Systems Biology, where we have the state-of-the-art sequencing and genotyping facilities, moderate computational infrastructure. I think many of you have already visited us. If not, we would like to invite you to work with us on the different aspect. So basically, we started to work on these different aspects. And I think while working together with large number of partners here in India, DGR and State Agriculture University, many partners in China, many partners in United States, Europe, and Africa, I think Groundnut community has done wonderful work. So for instance, in 93, they developed the first genetic map. Many of you know this is yellow tetraploid. You have two genomes A and B. So the first map was developed in 93. However, this took about 8 years to develop the first synthetic tetraploid map in 2001. I joined ICRISAT in 2005, and then we were very successful to develop the first map in the 2009, and basically based on the SSR genetic map, etc. But after that, we started to have a lot of progress, and now you can see the QTL mapping, GWAS, etc. And I think 2019 was a significant year where we had the 3 reference genome of cultivated tetraploid species. We started to have the molecular breeding products, and now the similar kind of things are moving further. And we will discuss in this presentation that how and in which direction Groundnut community can move further. And this is some significant example of this collaboration work. ICRISAT feels privileged to work with large number of partners around the world. As I said, different countries, and we had these papers for these both genomes, A and B genome in Nature Genetics. We had the A genome in PNAS, another B genome we had in the Frontiers, and then there was another paper in this giga science. And then in this 2019, we had this tetraploid genome, and this was again together with Dr. Bijian Zhuang, and this was another with the US initiative. This was another with the Dr. Xiaoping Chen and Dr. Liang. So I mean, there are different initiatives which deliver the genome sequences for these complex crop like that one. But things have not stopped here, and community is moving further, and I will discuss further that how we are doing. But the other thing is that once you have the genome, and from the plant breeding perspective, you would like to see that, well, how we can do this, basically make the application of genomics in the breeding program. For that, you need to have the marker genotyping platform. Earlier, community was using just SSR markers, but now, even when I joined ICRISAT, we used to have only just 200 or 150 SSR markers, but then we started to work with the community. Now, the good news is that we got large scale SSR markers, we got the DART platform, CASP marker, genotyping by sequencing platform, high density genotyping platform, whole genome resequencing, and now we are going for the mid density, low density, because we say one size does not fit all. So for application, depending on your application, what you need, you can use those kind of platform. Here, this is the example of having the high density genotyping assay, where in the single experiment, you can generate the data for up to 56,000 SNPs in a single go. Earlier, people were taking a lot of time. Now we have this possibility, GBS, whole genome resequencing, and our group and many other partners, they are working using all these technology very extensively. Now, we are also working right now to develop the mid-density genotyping platform. For instance, now when community wants to move towards the genomic prediction, and those high-density genotyping array are a little bit expensive, like $50 per sample kind of thing, but we would like to reduce this cost. We ask the question, can we bring the genotyping cost, for instance, $8 to $10 per sample? So with this kind of perspective now, we are going for that 5,000 SNPs array, and this is being in advanced stage. Now further, the low density marker based platforms, for instance, when you are working on the marker assisted selection, you just need few few markers. So for that one, 10 markers to 48 markers. For these things, we have developed the other platform. And now my colleague Manish Pandey, together with other partners here at ICRISAT, is in the advanced stage for developing these QC panel for the groundnut. So I mean that, so basically we are having a range of the genotyping platform. And as I mentioned, so this platform is being used with the breeding material of ICR-DGR, US Dharwad, RARS Tirupati, and of course with our partners in ESC and WCA. So those kind of things are available right now. And as this topic of this presentation was that, well, what we can do now in this genomic era. So basically this is a very important thing. And again, the breeding applications, they are our first priority, that how we can enhance the precision and efficiency in the breeding program, that too at the lower cost. So to address this issue, we think that now this is the time for the translational genomics. So not the genomics, rather translational genomics. What does it mean? How we can translate the genome information, the breeding program. You can see that we have this fantastic colorful genome, but we need to address the issue of the farmers on the right hand side, which you can see, and we need to keep the farmers in our mind that what we need to do. So for doing these things, we need to do something in the middle by using the genomic innovations, plant breeding innovations, together with other disciplines, including physiology, pathology, entomology, different disciplines. And in the end, we would like to translate this genome information in the breeding program, and we call it translational genomics. Now, if we discuss with different people, we can have the different kind of a strategy. This does not mean that this slide is having all the strategies. Huh? So these are a few which we discussed in the recent tag paper. Several of you were already co-author on this paper. So here we need to have the different kind of genetic resources. Nowadays, very important thing is that we need to develop a specialized mapping population like multi-parents, advanced generation intercrosses, nested association mapping population. And now groundnut community has already developed those kind of populations. We already have the germ plant set. Now from the genomic resources, we got the reference genome. Now people are moving towards the pangenomics area. We are resequencing germ plots. We need to develop the gene expression atlas. We need to have the candidate genes and markers. And then we need to address all these issues, what we need for the breeding program, for the different kind of breeding approaches. So if we ask the question, so what we need to do now? Well, you can do a lot of things depending on your objective, depending on your resources, time, etc. But I would like to list a few things here. Again, as I said, this does not mean that you all need to use all the approaches, or you need to use